Hello, Prof. Su. Mhm.
Thế hả?
Hello,
Hi.
Prof. Su.
Hi, hello. Yeah.
Yeah,
Okay
yeah,
now?
we can hear you. Yeah, we
Good.
can hear you.
Okay, let me share the screen just a moment.
Prof. Su, I have to introduce you first. It's okay.
Okay, just a moment. Yeah.
Okay, so selamat sore. Assalamualaikum warahmatullahi wabarakatuh. Itu adik-adik yang telah bergabung dan juga mungkin ada beberapa rekan peneliti juga yang sudah mengikuti. Ya, ada juga beberapa rekan peneliti ini ya yang juga ikut tergabung di forum ini. Thank you for your attendance today. So before Prof. Su starts his lecture, I would like to give a brief introduction for Prof. Su. So Prof. Su is the director of the Third Institute of Ocean. I'm sorry, he's a deputy director of Engineering Research Center of Marine Biological Research Comprehensive Utilization in the Third Institute of Oceanography, China. Jadi institut ketiga ya. Di ini badan risetnya langsung di bawah Kementerian Pengelolaan Sumber Daya Alam. Jadi mungkin seperti sekarang yang ingin dibentuk oleh BRIN. Jadi memang lembaga riset penelitian. Tapi I think Theo, singkatannya Theo itu, Theo also can work together with Xiamen University for accommodating postgraduate students. So when I visit Prof. Su in China, there were some international students and some were from Indonesia and also from Africa and also some were from Egypt. So Prof. Su, I would like, hello, how are you? How are you doing?
Fine. Thank you.
Okay.
Thank you for your
Thank you very much.
invitation.
Thank you very much for your time today. So your presentation should be around 60 or 90 minutes is okay. Please start your presentation. Can you share your screen?
Okay, now
Okay.
going.
Yeah, we can see it.
Okay. Thank you again for your inviting me to give this lecture for your colleagues and students. Again, I am from China and I work in the Third Institute of Research of Oceanography. And very nice to be here. And I will use about 1 hour or 1 hour and 30 minutes to share some of my research background and my research achievements with you. Okay, so let's go. This is my presentation outline, and I will introduce from the environment of marine and also introduce some marine organisms living in extreme marine environment. And the second is the collection of samples from those extreme marine environment. And the third part of my presentation is the screening of functional microorganisms. And follow this part will be the identification and the valuation of bioactive compounds. And the final part of my presentation will be the exploitation of those resources. I will give you some example. How do we use those resources of microorganisms from marine extreme environment? Yeah, and before we go into my presentation, I would like to give you 3 very basic background. The first one is that the application of microorganism, and it is bioactive compounds against the disease in human, in animal, or in plant is for a long time. It means that we have used microorganisms to deal with our disease for a long history. Okay, this is the first background. And the second is that there are some very important medical properties of microorganisms, such as antiviral, inflammatory, anti-allergic, and antioxidant. And those properties of microorganisms can be used to reduce the risk of many diseases, like cancer, like characters, respiratory issue, also some of the diabetes, including the Alzheimer's. And the third basic, okay, I'm sorry, and the third basic knowledge is that normally that we can collect those microorganisms from land, but through our research, and we found that we can also collect those microorganisms from marine environment. And we find that in marine extreme environment, we can collect more functional microorganisms. Yeah, it means that in marine extreme environment, there are more microorganisms resources which we can use for medical utilization. Yeah, so that's here below. I want to introduce some of exploitation of microorganisms from extreme environment. Extreme environment. This is the marine extreme environment. Yeah, and we will focus two types of deep sea in the polar region. And this is deep sea, and this is polar region. Polar region including Antarctic and the Arctic. We have two polar, yeah, we have two polars in the world, and this is deep sea. Okay, this is deep sea, and the environment in deep sea is very strange. Yeah, for example, very dark, no sunshine, no sunlight, very dark, and very salty, and also very high pressure. Of course, very toxic. In the xenobiotic also, for example, high sulfate, heavy metal. This is the environment in the deep sea, and deep sea. And this is the creatures, animals or microorganisms living in deep sea. This is the hydrothermal vent, yeah, and the deep sea. There we can find the temperature can be as high as more than 100, even 200, even 300. Very high temperature around the hydrothermal vent. And for the polars, this is the Antarctic and this Arctic. For a polar region, that's the environment there also very tough. For example, very cold, very low temperature, and very little sunshine. And also nearly zero annual precipitation. No raining. Almost 1 year, no raining. Okay, so we can say that this is a white desert. Very dry. Yeah, even a lot of ice, but no raining. Almost every year, no raining. So this is a desert, Antarctic. So the environment condition there is very terrible, very terrible. Good. So here comes one science question. Yeah, you know, we are doing the science research, so we like to give the science question. So in those extreme environment, that's people is very easy to come up with one question, science question. That is, how does those organisms can survive in those extreme environment? It means that how can those organisms can survive in very hot temperature or very cold temperature, very toxic, very high pressure, it's so harsh and so tough condition. How can those organisms survive? So this is the science question. We must answer to this question. Okay, so that's okay. So through research work, and the scientists find some answer for this question. How can they survive in those very poor conditions? The first answer is about the survival chemistry. That's survival in high temperature. Scientists find that inside those organisms, inside their body, that we find one kind of, we call the trehalose. Yeah, this is one kind of polysaccharide, trehalose. And this kind of trehalose can keep a protein from denature. This is the trehalose. And if your body, inside your body, if you contain this kind of material, this kind of compounds, trehalose, then that this kind of material can keep your protein from denaturing. We know that if your protein and high temperature become denatured, that it means that the cell will be died. So this is one kind of survival chemistry. And the second material, second compound, in fact, this is one kind of enzyme. And the scientists call this enzyme, we call it extremozyme. And this enzyme is very special. And this extremozyme can keep the cell work effectively. You know, in very high temperature, that's the cell, our body will become very hot, and they cannot take into function. Okay, and but this kind of enzyme can keep our body, can keep the cell with very good working condition, So this is another technique for those kind of organisms to be survival in high temperature. And also, now another phenomenon is how to survive in low temperature. You know, in Antarctica or Antarctica, even under the deep sea, we find that some of the area, the temperature can be as low as -80, -70. You know, for our human being, if the temperature is lower as -40, even 45, and for human being, we can only stand for less than more 30 minutes. Yeah, 30 minutes, and we will be frozen, and our cell will broken. Why? Because in low temperature, this we call the ice crystal will be produced. And when crystal produced, you know, this is the shape of the crystal, ice crystal, very sharp, very, very sharp. So if inside our cell, this crystal formed because they have a very sharp peak, and they will make the brain, the membrane of cell broken. So this ice crystal, sometimes we call this the flower of death. So it means that if we put into the very low temperature environment, -40, and after 30 minutes, inside our body, the ice crystal will be produced, and with their very sharp peak, that our cell will be broken, will be broken. Okay, so so this low temperature that we found that some organisms in Arctic, in the Antarctic, even under the deep sea, that they have this ALPS, means anti-freeze protein. Yeah, they have this kind of protein. In this protein, this kind of protein, they can avoid, they can prevent this ice crystal produced. It means that if we have ALPS inside our body, and no more ice crystal will be produced. So this LPS can provide us from cell broken, And in my neighbor, that we have found this LPS. And in my neighbor, we can produce this LPS. And we also use this LPS to, and we inject this LPS into the fish, and then we put this fish into the refrigerator, or we put this fish under the temperature -20, and we really find that this fish can survive because LPS will protect them from frozen. Okay, and another survival chemistry is we call one carbon chemistry. One carbon chemistry means that some of the animals, some of the organisms, they can use this one carbon chemistry technique, one carbon metabolic pathway to survival. Yeah, for example, that they can survive in very toxic. Very toxic means only carbon, and they can use this one carbon. This is one carbon. For example, this is, we call it, this is CH4. CH4 is very toxic, but many organisms living in the deep sea, they can use this CH4 to be the energy for their body. So we call this is one carbon chemistry, because they have this very special metabolic pathway. Later on, we will introduce some of the knowledge about metabolic pathway in some of the organisms later on. Okay. And that we found that in marine extreme environment, that many kinds of organism survival. Yeah, this one is we call the blind shrimp, no eye. Yeah, because it's very dark, and so for the evaluation, no eye on their body. Yeah, and also we find that some of the microorganisms also with a high abundance, very high quantity of microorganisms survival in deep sea in very toxic environment condition. And scientists, through research work, that scientists found that this microorganism in extreme environments have evolved unique way of survival, and which makes them possible to become valuable resource for excavating bioactive compounds. So this is the key sentence this afternoon we are going to discuss together. It means that some of the microorganisms living in marine extreme environment, that they can survive because inside their body they have some bioactive compounds, bioactive substance, and those bioactive substance is very useful for the human activity, So we can use these bioactive compounds to be the medicine for our human being disease treatment. And later on, I will focus some of my research work about those microorganisms in the bioactive substance utilization. Okay, now second part is my institute, my research institute, the collection of microbial sample from deep sea in the forest, from deep sea. Yeah, in my research institute, we will conduct the collection of sample from deep sea for nearly 20 years. For nearly 20 years. And we go everywhere in the world. We go everywhere to hunt this micro sample in deep sea. We go everywhere. And altogether, we have conduct 40 cruises in 20 years, from 2002 until now, about 20 years, for the collection of microbial sample from deep sea. And this is the facility or equipment for sampling, to collect sample from deep sea. We use those equipment. Yeah. I know maybe you know this equipment, this machine, that this kind of machine can go into the sea, down to the sea for 10,000 meters deep, 10,000, yeah, 10,000 meters deep, very, very deep. So far, here in China, we have two. We call this is the deep sea submarine. We have two. Yeah, for this one and this one. This one is around 6,000 meters deep. And this one can go down to nearly 10,000 meters. Very deep. Okay, so, and that's with this equipment, we can go everywhere to collect every kind of sample from the deep sea, So we go everywhere. Yeah, we go everywhere. That's in deep sea. So including the Antarctic. This is Antarctic. And this is Antarctic. This is Antarctic. Okay, Antarctic in the Antarctic. And between the north and the south, everywhere we go to collect this deep sea microorganism sample everywhere. So I told you that we have conduct nearly 20 years to collect samples. This means that we collect sample in Para region. We drill a hole deep to about 100 meters deep, and we collect the sample. And people said, after identification, they say the sample is nearly 1,000 years before, even 2,000 years before. Yeah, because this sample collected from nearly 100 meters deep in Antarctic. And we also go very inside the Antarctic and the Antarctic, very inside, very far away. And we collect some sample. Those samples also existing there long time ago, maybe 2,000 years ago or some years ago, very long history. And so far that we have collected nearly 30,000, more than 30,000 this collection in stock. In my research institute, you can find that we have a library to stock those samples from everywhere. So this sample is very valuable because based on those samples, we can conduct a lot of scientific research work and find some very important scientific answer. Yeah. Okay, the third part of my presentation is the screening of those functional microorganisms. Those functional microorganisms. Of course, this is for culturable trees. You know, some of the microorganisms from deep sea or from the Antarctic, in fact, is cannot culturable in normal condition. So this screen is just for the culturable stream. Culturable. It means that we can provide the normal condition in the naked survival in my neighbor. So we call this the culturable trees. Yeah. And before we conduct our screening, we needed to establish some model for our rapid screening. And so far, we have established here about the five model. This is 5 model. Yeah, we have established this 5 model. And we also construct 2 kinds of technology. One is the HTS. Another is the HCS. HTS means high throughput screening. HCS means high contents screening. This is the rapid screening model in the technology for our screening of functional microorganisms. Yeah, this is the high throughput screening assay in 96 well microplate. Here in my neighbor, this is almost automatic. So those kind of machine work every day in my neighbor automatically. And with this PS screening assay in my neighbor almost every day, 24 hours a day, automatically. So because this is a model, so we set up and we want them to work very hard day and night for our HTS screen. And this is the cell-based high throughput. Especially if we want some utilization compounds or microorganism, use this cell-based to do our HTS high throughput screening. Yeah. We use here in my neighbors many kinds of this tumor cell. Tumor is cancer. Tumor cell as the cell-based screen. This is my student, and now she's check about the screen result and try to find some good achievements. And we also use some animal. Yeah, this is the model organism-based high content screen, HCS high content screen. We use this zebrafish. Zebrafish is very useful in my neighbor because this fish is very small one, very small one. Yeah, and it's easy to be cultured. Very easy. They are very easy to survive. And they occupy a very small space. For example, this one, we culture 20,000 pieces of this zebrafish. And this zebrafish and can be used for high content screen. So far we use this model. We have harvested more than 100 bioactive compounds for medicinal utilization. So their working efficiency is very high. So for pharmaceutical industry, my neighbor, we also provide some service for pharmaceutical industry. In in terms of pharmaceutical utilization, they just want to obtain some medicine from my labor. So to provide this kind of service that's we set up our technology just for this pharmaceutical industry. For example, this is anti-cancer, anti-proliferative strains. We want to find the strains which can anti-cancer, and we also want to screen some of microorganisms which can produce good enzyme. Yeah. You know, good enzyme can be deal with some stomach, yeah. And also that we want to isolate some strains for antioxidant. For example, for cosmetic, yeah, people, especially for girls, they like some cosmetic for their skin, for their face, and we can provide this antioxidant. And with this antioxidant material, they can produce some kind of medicinal cosmetic for girls. Yeah, and also that we want to isolate some anti-inflammatory property. Yeah, it means that they can antibacterial. Yeah, they can prevent some inflammatory issue. So that's we screen for pharmaceutical industry. Normally, we conduct those research work and just want to find some biological antibiotics, immunoenhancer. Immunoenhancer means that those kind of material can improve their immunology, yeah, in the probiotics, and also digestive, in the antibacterial, in the anti-cancer, okay, anti-cancer, and also some infection treatment. So those is just for pharmaceutical industry, and this kind of job is the most important job in my labor, because with this kind of job, we can cooperate with pharmaceutical factory, pharmaceutical company to work each other to find some normal pharmaceutical drug. So in terms of this screen, and we can divide into the primary screen and the rescreen. Primary means that we want to find some property. In the rescreen, that we want to find what the exact properties. Yeah, for example, that this microorganism can anti-cancer, and we want screen again to find which cancer they can deal with. Yeah, you know, for human being, many kind of cancer. Yeah. So for screening, that we need primary and the second step of screening. Excuse me, maybe some of the problem with my PC. Let me check again. Okay, now, and I just want to give you an example about the screen of microorganism. Yeah. For example, that this is the screen of this kind of, this is bacillus. We call it MS2 bacillus. And we needed to do some sequencing of this strain. This is the 16 srDNA sequence. And we got this data. And we just put this data into the bank, NCBI. This is in the United States, NCBI. And through comparison, this bank will give us a number. So we can label ours with data. Okay. And then we also do some identification. And then we can give the scientific name to this strain. For example, this is Bacillus subtilis MS2. Okay. So this is the okay. Yeah. And we also need to do some complete genomic sequencing. Excuse me, okay, let me check. Yeah, I'm sorry. So this is about the complete genomic sequencing. Yeah, here in China, that's the complete genomic sequencing become very cheap. It's around, for one strain, it costs in RMB around 8,000. It's around, I think, 1,200 US dollar. Yeah, for one strain of this microorganism. So become very cheap, not so expensive. So I encourage my students to send this strain to do the complete genomic sequencing. Because with this complete genomic sequencing, and we can know the gene in detail, functional gene in detail, with this data, data from the sequencing company. So we know something in very detail. Okay. For example, if we have this complete genomic sequencing data, then we can put this data into database. Then through the comparison, and we know some of the functional gene in our strain, in our microorganism. Yeah, in our microorganism. Maybe you know something. But anyway, we will tell this basic knowledge later on. Even they can tell you what is the scientific name of your strain. You know, yeah. In this table, we have two strains, MS2 and MS22. Okay, this is two. And we put this complete genomic into database. And through comparison, we can find some result from this database. Yeah. So you know that we find some of the normal family. So we know the position of our strain. Yeah. Even we can find some of the innovative strain, innovative achievements from the comparison in database. Yeah. And also from this complete genomic, that we can find some of the core gene, some of the specific gene. Through analysis, we know some of the gene, for example, core gene and a specific gene. Yeah, such as that we can find that this kind of gene is just for I'm sorry. Yeah, for metabolism. Yeah, and we can also find the sample is for disease prevention, disease. Yeah. So this kind of gene, yeah. So we can find some of the, so how many gene? Yeah, this one for disease prevention, 3, 4, 2, 1. Altogether is 7. It's about 7 to control the disease happening. So it means that from this gene sequencing data, we can find some of good gene, functional gene, specific gene, point gene. Yeah. So this is about gene analysis. And this is the functional gene index. And we have this system, and this system can automatically to set up this index for you. For example, this is the growth promoter. CH, this is gene, CCK labeled. And this kind of gene is just for growth promoter. And this is AMP, is just for disease resistance. And this MX is for immunity stimulant. So it means that this is for reproduction, for maturation. So from system, that they can automatically set up this label for you. We call this a functional gene index. Yeah. So the sequencing science now is become very popular and become very easy to do. Yeah. Okay, and also that for our target gene, that because that maybe the target products, for example, enzyme, they can, for the original strain, they can produce very little in amount, and also not so active. So normally we need to do some cloning in the expression, in another system. So this is, we call it gene engineering. Yeah, and for gene engineering, we have three steps. The first step is that we need to establish, we call the establish our plasmid. We have need to establish this plasmid. And the second, that we need to put this plasmid with our gene, combine each other, and combine, this is our gene, target gene. And with this plasmid, we link each other. And then we just put this one into the E. coli, into E. coli. It means that this E. coli will contain our target gene. And then through the fermentation of this E. coli, they can produce our enzyme with high quantity and also very pure and also very active. So this is about the expression of this gene in E. coli, yeah, in my label. This one now is become easy to do, and also very easy to be successful. In this way, we can produce our target easily with very high quantity in a high purity and also very active. Yeah. So this is about, we call the gene engineer. I will introduce to you later on about the gene engineering. And also that we want do some prediction of our enzyme. What kind of structure? And we select from the data bank, we select these two. Other scientists, they have found that the protein, enzyme is one kind of protein. And they show us this kind of structure. And this is my enzyme for strain 2. And this is my enzyme for strain 2. In after comparison, we found in this is my product. My product is almost the same as this one, because this is a very important area. This is a very important area. We say we can call this the functional. We call this the functional area. In very similar with this one. Of course, this one is the same, almost the same as this one. So this we just say this is the prediction, not exactly. And this prediction is very important for the future utilization, because we know where is the functional part. Okay. And this is our strain library in my neighbor. So far in my neighbor, we have accumulated more than 20,000 strains, in my library. So I hope that in the future, I can invite you to use my resource. Yeah, I can provide those strains, and we can share those resources with you, with your university, yeah, with your neighbor. So those functional strains now is ready in my neighbor. Yeah, in more than 20,000 functional strains. We can regard this is a very valuable resource for our microbial research work. Okay, the fourth part of my presentation is the research and the valuation of bioactive compounds. You know, from the strain, from this microorganism, very important is to use their bioactive compounds inside bioactive compounds. This is important. Yeah, and before we use those bioactive compounds, first we need to do some extraction. We need to isolate these bioactive compounds from the strain, from the microorganism. Yeah, so and you know that some of the our strain, some of our microorganisms we collect from deep sea. You know, deep sea is very high pressure. Yeah, high pressure. So we have this equipment, and this equipment can provide high pressure for those microorganisms, and then we culture those strains, and in this way, that this strain will produce active components. Yeah, so this we call the high pressure cultivate. Yeah, those one. You know, that's we can adjust the pressure from 0.1 to 140 MPa. Okay. In the temperature, you know, some of the temperature that they need a high temperature. Some of the strain they need high temperature. So we can adjust the temperature from 0 to 135. We find that some of strains they can only survive more than 100 temperature in the boiling water. Yeah, very, very high temperature. But they can survive and they can produce bioactive compounds. Okay, and after cultivation, then this is the separate. Okay, this is the separate of active compounds. Yeah, we have this equipment. Yeah, and we can isolate, we can separate that those bioactive compounds one by one. Yeah, and after we get these bioactive compounds, bioactive substance, yeah, and we needed to do the partition, yeah, attributes partition in the identification of bioactive compounds. We needed to do this one. And this process including two steps. One step is that the foundation research of this effective substance. And the second one is the quality control. And we use this qualitative analysis. And also we use the activity assessment to do the research work of this foundation of effective substance. And for the quality control, we use this quantitative analysis and also the limited test, because in those kind of products, maybe they have some toxic products, and we need to test which kind of toxic and how much these toxic components inside. We also need this one. Yeah. And this quantitative analysis is just for the effective constituents. This is our target product. And this is not what we want. We want to limit those kind of toxic products in our target products. Yeah. So give you example. You know, that's for example, when we get this extract in a this kind of total extract, and we can divide into many groups, B1, B2, many groups. And then in this B1, we can also divide again to antioxidant. In this B2, maybe this anti-inflammatory. And maybe this one is to cancer, anti-cancer. And maybe this one is for some other function. So from total one, and then partition to many kinds of this kind of material. So in this way, we call this is the attributes partition of our bioactive compounds. And this is the technology we use to qualitative analysis. Yeah. I think maybe in your university, you also have this equipment. This is the LC Q-TOF mass. This is LC IT mass. This is NMR. With this three kind of equipment, then we can know the structure of your material or your target products. This just give you an example about how to identify your effective substance. Yeah. Through this LC mass, QT-TOF, and we know the molecular weight. This is the molecular weight, and we know this is the molecular. Okay, and we know the structure. Then we know, oh, this is glucoside. This is one kind of glucoside. Yeah, we can name all your products. And we can also know that's how much the percentage of your target products in your combined products. And we also use that's the negative model and the positive model we call the cross check. In this way, we can get the results of identification. Yeah, this is the achievement of one of my PhD students with less than 1 month. And he can identify so many kind of products with the exact structure and also the function, anti-cancer or antibacterial or antioxidant. Yeah. So this is about the qualitative analysis of bioactive compounds from microorganisms. Okay. And then we come to the evaluation of bioactivity. Yeah, you know that we have these bioactive compounds, but we must know the activity, how high is the activity. So we needed to do some evaluation about their bioactivity for our components, And we use this, we just, for example, this is about the inhibition, antibacterial. And we put every kind of our bioactive compounds, and we find their ratio of inhibition. For example, this one with very high activity against the bacteria pathogen. This one. So anyway, you can find some of your products with high activity. For example, this one, and this one. And the result can be automatically come out by computer, very easy. So you just print out the result from computer. They can tell you the answer with kind of components with high bioactivity. For example, that's we can find that from this data. This is about 40, 31, and this is minus. So of course that this ranking 3, ranking 15. This ranking 3 will be our target product. So, and for this kind of research work, so far we have accumulated many kind of bioactive compounds. We have so many kind of, and you can find that this data is automatic. So, for example, today the students put one product inside, in the computer will show how many kind of products. And with this label, we can scanning, we just scanning, and we can know every information about these bioactive compounds. So far we have this natural product bank. We have, and again, I invite you to make use of those resources together with my label. Okay. So far we have more than 13,000 fractionate second metabolite extraction from over 400, only 400 microorganisms. So we also have this bank for natural products. The final part of my presentation will be the exploitation of microorganisms or microactive compounds. Okay, nearly 1 hour, but anyway, I will finish in 30 minutes. Okay. So as you know that we want to use those microorganisms or bioactive compounds, and we will use 3 kinds of material. The first one we want to use is the gene. We want to use the gene. Okay, this is the gene. We want to use the gene, and we want to use the bioactive compounds. And also we want to use the microorganism. So these 3 labels we want to use from the microorganism, bioactive compounds, and also the gene, because some of the microorganisms cannot survive, we can only use their gene. That is that we want the gene, bioactive compounds, and the microorganism. And to use the gene, I'm sorry again. Use the gene means that for some of the strain, unculturable strain. In this way, we can only use their gene. So this is the approach for culture independent we cannot culture this tree. Maybe we cannot provide a good condition, very high pressure, very high temperature, okay? Or so we can only use it as in. In this way, I won't discuss with you about the gene engineering. And so we have these 3 steps to use the gene. First is the metagenomics. We use this technology, we call it metagenomics. And we use this metagenomics to find the good gene. And then that we use the gene engineering I just told you about this good gene. And then we select this good gene. And then with this gene engineering, we produce our products with this gene. Okay, in another system, for example, in E. coli or in yeast. Yeah, because E. coli we can culture, but we won't use this gene. Okay. Yeah. Metagenomics is in fact that is very powerful. Metagenomics. Okay. Metagenomics is the study of genomic content that we covered direct from the environment sample. Okay. So we have these 3 steps. First step is to extract the DNA of all microorganisms from the environmental sample. In the second, construct a metagenomic library. Okay, and then conduct our research work to find their genetic composition, and their community function, their physiological active substance. Okay, in fact, this metagenomic just provide us to find the good gene. Okay, so that's the basic types of metagenomic studies, including that first is we needed to do the sequencing. I have told you the sequencing. And from sequencing, we know some of the good gene. Okay, so that's we will find this good gene. This is a good gene. If we say this gene is very normal, it's a very normal gene, very innovative, then we can use this one. Okay, so those work must be based on the sequence, and we have this tool to do this analysis automatically. So you don't worry. Yeah, you don't worry. You just send your sample to be sequencing. Then, and with the tool, you have some tool that the sequencing company provided to you. And this tool can do the analysis automatically. And they can give you the result very quickly, to find this normal gene, innovative gene for you. Okay. And second is about the genomic expression in another system. Yeah, for example, in E. coli, in yeast, in another cell. In my label, that we have 3 kinds of systems to express. One is the E. coli, the second is yeast, and the third one is the algae. Some of the single cell algae. We can express. You provide a good gene, and we can combine your good gene into algae, into yeast, also into some bacteria. And of course, that not only this genetic, but we also need some pathway. So we also need to construct a pathway for your gene to express our target products. Yeah, this is about the pathway. And those pathways now also very easy to find out. Yeah, very easy to find out. Because the time limited, and I cannot tell something in detail about how to find this metabolic pathway inside the cell. Yeah. But anyway, this information is very easy to master. This technology also not so difficult to master. Yeah. And this, the final is the gene engineering and also synthetic biology. Yeah. So that's first you need to design a model in the compose functional unit. Functional unit means that your gene and also your pathway together. This is the functional unit. In a assemble unit, assemble your functional unit into yeast, into bacteria, into algae, yeah, into, yeah. So this one we call the underpin cell. Underpin cell. Underpin just like how to say support cell. So you need to compose this functional unit and assemble your unit in a line, just like in factory in a line, and then that combine your working line into your cell, your single cell. Then you can make them to go together in exactly the right order in the orientation. So you just turn on the gene, turn on, and then they can automatically to produce your target products. This way we call it as synthetic biology. Yeah. So this is the underpin cell. Okay. So synthetic biology, in fact, that's you need connecting your gene into a network and allowing your gene to work quickly, to work effectively by your policy. Okay. So this is about the working line. This is gene. Yeah. This is the gene. Yeah. You put it inside. Okay. So gene circulate, enzyme, metabolic pathway, all together. This is the working line. And now we call this line every link. We call this the biobricks. One brick. Just a factory, yeah. And then they can automatically to produce your target products. And also that this factory can learn by themselves. Design, build, test, learn. Yeah. So they can accumulate their knowledge, their experience automatically. So we call this the study machine. It means that your machine can start, can improve by themselves. Yeah. So we call it DPTL circle. This is the circle. Design and then build and test and learn. So this is the circle. So if you set up this factory, and after maybe 1 or 2 months, that their working efficiency can increase because they learn every day. They do the circle every day, so they can improve themselves. Okay, so this is about this metabolic engineering in the metabolic pathway. So we just quick to have review. This is the construction of biosynthesis plant, just the factory, yeah. Just this kind of factory. Okay, quickly, yeah. So this is gene engineering in the synthetic biology, quickly to go. And I want to give you an example. Example demonstration. In my lab, that we find this product we call it BKE7, and we find that this BKE7 can be used to treat this leukemia. Yeah, leukemia. You know, leukemia is a very serious cancer. And this kind of cancer is very easy to find from the children younger than 15. Yeah. And we find that this BKE7, these products can deal with this leukemia. Okay. And we find that through this model, many kind of products, and we find that this kind of products is very high efficiency to kill this leukemia. Yeah, you know, this kind of products. Okay, and also that we found that our products has a very good relation with time and with dose. Very good. For example, 24 hours, 48 hours, 70 hours. They have a very good linear relation. Okay, and we can find that disappear. Yeah, very good result. Yeah, really. And now we contact with one pharmaceutical company, and we will develop this kind of products into a medicinal drug. Yeah, we hope we will be successful in the future. And also that how to use this functional tree. Yeah, cultivable. That demonstration is that we use this kind of tree for this ecological and green agriculture. We select some of these functional tree. Yeah, functional tree. We altogether we have 4. Yeah, this is one, and this one can have ability strong antibacterial, and this one can have a stronger enzyme activity. And also this one, they can produce immunopolysaccharide. It means that they can produce this polysaccharide to improve your immune ability for the fish also. In this one, it was strong degrading of nitrogen. Yeah. So, and we use this fermentation to produce these microbial products. Okay. So altogether, we produce these 4 products because this is used for aquaculture. So very easy to get permission to go to the market. Yeah. Even for human being, we have a lot of regulation, as you know. But for the aquaculture, for the fishery, very easy to go to the market. Now our products have go to the market successful. Yeah, and they have been used for aquaculture, for the shrimp aquaculture, they have been used. Yeah, and we have got a good harvest, very good harvest, very clean, very healthy. We never use the chemical drug. We only use this microbial products. And now we have set up the relation with a few farmers, with 2 or 3 farmers so far. And we just provide these products, and they use our products for their ecological aquaculture of the shrimp. Very successful. Okay, summary. Yeah, that's marine extreme environment is a variety of microorganisms which contain bioactive compounds and can find a good application. Okay, in my lab, so far over 20,000 these functional trees were harvested from poles, Antarctica, in the deep sea. In the morning, 13,000 these extraction were prepared, and hundreds of compounds were obtained. So, in those products has a good property for anti-tumor, anti-flu, anti-pathogen in the immunity enhancement. So I hope that we can make use those resources together. Okay. And final is my suggestion for cooperation. I also discussed with Professor Eka before that I want to set up some cooperation with your university. Yeah, I have three suggestions. The first is about the students exchanging. TIO, my research institute in the your university can exchange the students each other. In my research institute, I can provide many scholarship for you to pursue master or doctor degree in my research institute. So in the future, I really hope that some of students from your university can come to my research institute for master degree or for doctor degree. Yeah. And the second suggestion is exchange scientific researchers to work in my lab for 3 months to 6 months for joint research work. This is my second. I can provide 3 to 6 months for you to be working in my lab. And the third suggestion is that we can apply some of the project each other, international cooperation project each other. You know, China, we also provide some opportunity of the application of cooperation project. So that's I hope that in the future we can apply jointly. We can apply those international cooperation project each other in the future. So I have this three suggestions. This is my research institute. Okay. I think Professor Eka know my research institute because about 3 years ago, I invited Professor Eka to visit my research institute. And my research institute is located in Xiamen, a very beautiful coastal city here in Xiamen, in south of China. Okay. And this is my neighbor, my research center. I hope that if the pandemic is over, please visit my neighbor. Yeah, I will invite you to visit my neighbor. Okay, and I hope that Professor Eka will come to my lab again together with his colleagues, his students, to do some research work together. And you know, I very often we invite some of the research scientists to visit my neighbor, to visit my neighbor. They like to join us for conducting some research work. Okay. And I also, you know, I visit Indonesia very often. Very often. I also that this is around 2018. 2018, 4 years ago. You know, I am the member of these Pisces, and we set up a good relation with your BPPT, and so every 2 years I visit Indonesia to conduct this project in your country. This is my partner from BPPT, my partner. I went there to do some investigation with the local people. This guy is from Japan. This is from Canada. This is we are the colleagues in Pisces team. And we have some project, international project in your country. Of course, about the marine project, yeah. And the sightseeing, yeah, I like very much about it. Okay, so 1 hour and 30 minutes. So this is my presentation. And in my presentation, I just discuss with you about the utilization of microorganisms living in marine extreme environment. And the minority is that marine living in marine extreme environment has more opportunity to be used in medicinal industry. Yeah, and we can try our best to find some bioactive compounds for agriculture, for medical industry, even for our human being, for our environment. Anyway, in the future, I really hope that we can join each other to conduct those research work and try to find some good products to be used in the society. Okay, this is my presentation, and thank you again for your coming.
Okay, thank you, Prof. Su, for your presentation. And I would like to first welcome some students who would like to ask Prof. Su questions. You are also welcome to ask in Indonesian. Yang mau bertanya silakan pakai bahasa Indonesia. Nanti saya akan bantu komunikasikan dengan Prof. Su. Okay, there's one raising hand. So, Ardiansyah Satria. Okay, please address your question too.
Okay. First of all, actually, I want to say it was an interesting and also fascinating presentation from you, Professor Xu. And I actually have only one question for today, and my question is about a rapid screening test. During your presentation, you're mentioning about a certain model, such as a pathogenic model, animal model, or even a cell model. Is there, or are there any certain advantage, or is there any, I'm sorry, are there any a more effective model than the others, or every model is actually the same one and also have their own advantages? Thank you, sir.
Okay, I know your question, very good question, but because of the voice, I cannot catch the exactly. But I know that your question is about the screen. And in my lab, I have established some model for rapid screen. Yeah,
Yeah, precisely, sir.
I know that you are interested about this rapid screen. Yeah, but I cannot catch the exact meaning what your question is. I'm very sorry because the voice is. I just catch some about the screen, about the model, about the rapid screen. So,
yeah.
Okay, I'm so sorry. But anyway, please say about your question. Is it about the screen?
My question is about the model itself. So during your presentation, you're mentioning about a certain model for a rapid research test, such as pathogenic model, cell model, and also animal model. You're mentioning about zebrafish model. And my question is about, is there any, from this model, is there any certain advantage, or is there any much more effective model for rapid testing, or every model is actually the same one and have their own advantages? Thank you, sir.
Okay. Of course, we can help. So far in my lab, we have established, we have construct some model, and US statistics also very important. And we can, in fact, it's easy for my lab to set up another kind of model. No problem. Yeah. based on our purpose, we can set up different model. So if we want to set up some model, we must base it on our purpose. For example, that if you want your product, your target is to be used, for example, for the environment, yeah. Then of course that, for example, you want some of the microorganisms, they can absorb carbon dioxide. Okay, carbon dioxide, CO2. CO2 now is, you know, we don't like CO2 too much. In the suppose that you want to select some microorganism, they can absorb carbon dioxide very quickly. In this way, we can set up a model. In this model, just to screen the microorganism which can only eat carbon dioxide. Okay, so a very good question. Yeah, it depends on your purpose. And very easy to set up this kind of model. I hope that in the future that we can discuss by email. Yeah, by email. I can show you the email, and then I can give you the answer more detail. Okay, thank you for your question.
Okay, you're welcome. So, actually, I'm the one actually need to say thank you, and I was actually looking forward for working together or maybe discussing more about marine pharmaceutical industry. And once more, I will say thank you, Professor Xu.
Thank you again. Thank you also. Thank you. You are very welcome. How old? Excuse me, how old are you? Very younger, yeah?
Actually, I'm still on third semester, sir. I'm actually in my sophomore year.
Very younger and a very handsome boy. Yeah.
So please directly ask Prof. Su. Any questions anymore?
Any question? No problem. Any question? Yeah, we can.
Yeah. So, Prof. Su, I remember visiting your lab, and I never shared the photos. I'll share with you later. So 19, I think. Okay, Zain, you want to ask Prof. Su any questions?
Yes. Thank you, Dr. Eka, for the time for giving me the time for asking my question. And I would like to thank you very much for Prof. Su that already explained us about a great presentation. My question is, one of your slides was explain us about a comparison between strain MS2 and MS22. And my question is, I'm so sorry, could you please explain us more about MS2 and MS22, because I didn't really get any specific information from this. And my second question is coming from your from the good compound, sorry, for the from the active substance that's coming from living organism that you take in the deep sea. Did you is that sorry, for good application prospect, did you find any active substance that don't have any good prospect in the from the living organism itself, or just about don't that don't have any application for the human being? Thank you.
You mean from deep sea for human being?
Apa dia bilang, guys?
Hello? Excuse me? Yeah.
Sorry.
Sorry. Okay. A lot of noise. Yeah. When you speak, a lot of noise. I catch some meaning. You mean that I collect some sample from deep sea And you want to know if some of the strain will be good for human being?
Yes.
Yeah,
Yes.
so far, no. Excuse me. Okay. In my neighbor, so far, we have more than 20,000 microorganisms from deep sea in the polar region. In the among these 20,000 microorganisms, 400 microorganisms have been investigated. And among these 400 microorganisms, we have found 3 microorganisms, only three strains will be very good for human being. One is for leukemia. In my presentation, I say one strain, we find the BKE7, this kind of products will very good for the treatment of leukemia. Leukemia is one kind of blood cancer. Yeah. So far in my neighbor, among these 400 strains, we have found three microorganisms, 3 strains, which will be good for human being. One is for leukemia. One. And another two is very good for antioxidant. Antioxidant, it means that, for example, for cosmetic, also some of the, aging elder people. Why people can get old? Because inside our body, that because of the environment, because of the not so healthy food, so we needed to do some antioxidant. For example, if you put your face, your skin into sunshine, that's inside your skin, which will be a lot of oxidant. And this kind of oxidant is very harmful for our skin, for our organ, inside our organ. So we needed some products which can antioxidant. So among these 400 strains, we find that another 2 can produce very good products which has a very high ability to be antioxidant. And one of the antioxidant we want to do for cosmetic, for your skin, even for girls, they like some skin and some cosmetic for their skin, for their face, to prevent this oxidant. So oxidation is very harmful for our organ, for our skin, for our. And another strain with this antioxidant property, we will use for our stomach. We want to produce one kind of products for our consumption, digestive. You take some of the not so healthy products, and you will feel stomach. And we want to produce some capsule with these antioxidant products. So I answer you that we have 3 microorganisms. One is for leukemia, another one is for cosmetic for our skin, and another one is for stomach. So I hope that you can join us to find more microorganisms from my neighbor, from my library for human being. Okay, thank you for your question.
Thank you very much, Prof. Su. I actually want to add something to that. So I noticed that you also developed the uncultured, the microorganisms which cannot be cultured, but you also managed to develop products from it. You screen the metagenomic profile, and after that you express in expression system, and then you just produce the genes, the functioning genes.
Right.
Yeah, that is very interesting, because I didn't know that you can utilize the unculturable bacteria.
Yeah. You know, that this is very interesting story, you know, that we have got one gene in one of the microorganisms. And this gene is very useful for our eye. You know, in the deeper sea, very, very dark. Very dark. But we really find that some of the gene, they can catch the light. They can catch the light. So this kind of gene that we use the expression to produce some products. And we find these products is also very good for our eye. You know, now we have many problems, especially for younger people, that their eye, their ability to catch the picture, to catch the light is very weak. So we want to produce some of the products by expression with this gene. Of course, this gene we use the metagenomic. So for uncountable stream from deeper sea, that really we can find some very good gene. And through expression, through gene engineering, we can produce some of the very special products for our human being. Of course, it is not so easy, you know, because we find this kind of gene outer link to the E. coli or to some algae, and they become very weak to express. Maybe because of our condition in you know, not we cannot provide the deeper sea environment condition for this gene. So in my label, we also try to, how to say, to omic, to just to provide some of the condition, just like the deeper sea, and to support this gene to express the target products. But anyway, first of all, we won't find this gene. In the second, we'll provide some condition for this gene to express our target products. This is not so difficult, especially, for example, in my label, we have found many kinds of genes, but after expression, we find that nothing come out. This gene doesn't work. And the scientists say that maybe because of the condition we provide, not the similar as the deeper sea. So for the development of this normal gene, this good gene, we need two steps. One is the hunting of this gene by metagenomic. And the second is we also need to provide some good condition to support their expression. So not so easy, yeah, not so easy. But anyway, we have this resource. So through our efforts, maybe in the future, we can find some good methodology for our purpose. Thank you.
Yeah, thank you. I think we have more questions from the students. Okay, anyone can go first.
Okay. First, thank you very much to Prof. Changan Xu for the lecture this afternoon, and also thank you very much to Dr. Eka Prasedia as moderator in this session. First, let me introduce myself. My name is Hasriaton Fatmi. I'm fresh graduate from Biology Department from Mataram University. Now my affiliate in Dr. Eka's lab in Bioscience and Biotechnology Research Center. Now I'm looking for master scholarship, and my question maybe about the scholarship that you offer in the lecture. Now my field research in PUBB Research Center in molecular docking study, especially in from macroalga compound docking to several in medicine field, and also I'm working in macroalga extraction compound. From my background, is it possible if I apply the scholarship Prof, and how the procedure? Okay, I think that's all. Thank you very much, Prof.
Yeah, your voice is very weak. Professor Eka. Hi, Professor Eka.
Yeah. Well,
Yeah.
I think she asked about the
Repeat it.
study chance. So anyway, I would like to share just before we go. Farmasi, I have some photos of you. Time. So this one actually we're from with Prof Su, where we visited his lab in 2019, and we already actually discussed collaborations for student exchange. I think Prof Su, but because due to the pandemic, we have to postpone. So one of the Prof Changan Xu patents already also used in Jindan Nanotech. I think if you all know that, if I'm not mistaken, Prof Su told me that the Nike antibacterial T-shirt, it uses the nanofiber product from Changan Xu, which has nano antibacterial activity. So if you all buy the antibacterial T-shirt from Nike, that is one of the innovations are from Prof Changan Xu. And this was a lab, and I think I don't know if you remember this student.
Good.
This is Yogi. Yeah, he's now doing master studies in Bogor University. So Yogi spent around 2 months in Xiamen, but I think he was in Prof Chen's lab, not in your lab. So I hope in the future we have opportunity. I will send the photos later to you. I think in the future we should discuss again the opportunities for our side, because I think you previously said that the devices like LCMS and NMR is very essential if we want to investigate natural products, but we don't have that. It's very limited. Even in Indonesia, the basic LCMS, HRMS with electrospray, the Orbitrap. I don't know if you know, Prof. The Orbitrap, the LCMS a tandem with HRMS, the Orbitrap. We only have 2 in Indonesia, Prof. One in Gajah Mada University and one in National Agency and Research, which is in Jogja. So only 2 in Indonesia. I think one more is in Bogor University, free. So not so many. So if there is a possibility of sharing resources, it is very good from our side.
Okay, good idea. Yeah, I also hope that in the future, some of your students will come to my lab for study or for research.
Yeah, thank you very much.
Is welcome.
I think we only have only one more question. So, Fitriani?
Yes. Okay, thank you, Dr. Eka, for the time.
Thank you for the explanation, Professor Changan Xu. I'm Viviani from Mataram University. I want to ask about anti-proliferative strain for cancer and infection treatment. Question number 1. In this compound only found in extreme microorganism or all extending microorganism may be mesophil? If yes, why could this happen? Question number 2. How is the process until the compound can be used applied to cancer and infection treatment? Thank you.
Okay, yeah, this is the too big question, very too big question. Okay, but I hope that you know this is very complicated. And can I answer you by email? Yeah, by email. Maybe I will find the time by email to answer you. You know that I just attached my email address to everyone. Yeah, so I hope that I can answer you by email. In this way, I can tell you more and I can give you the answer in detail. Okay, you answer because that this system, this afternoon, that this system is not so clear. So I cannot. For me, it's very difficult for me to catch your meaning about the answer. But I know this is the too big question, very big question. So I hope that I will answer you that by email. Okay, please send me your question by email to me. Okay, thank you.
Okay, thank you.
Yeah, okay, thank you. So I think that closes our discussion session, Prof. Su. So, jadi mungkin untuk pertanyaan lebih lanjut itu Prof. Su juga sudah share email. Jadi beliau juga mempersilahkan kalau mau langsung email. Begitu. So I think we are running out of time, Prof. Su. Probably we could continue in the future discussions for our so one of my friends is actually also interested in your work. He also doing similar work. He's from the National Research Agency of Indonesia, and I told him about our meeting, and I think he will be interested in further discussions.
Okay, yeah, good idea. Yeah, I agree. Yeah. Okay, good.
Silakan deh di share sertifikatnya. Ayo, Prof.
Yeah, in fact, in the future, like.
Yeah. So, Prof. Su, we have a certificate of appreciation, and hopefully, in the future, we could invite you to Indonesia because currently we are also still adapting post-pandemic, and probably in 2023, hopefully, we have a chance to meet in.
Yeah. Good. Thank you so much. Thank you.
Okay.
Very great.
Silakan operator ditutup atau bagaimana di sesi.
So many people.
Before we ending this session, to all participants, please turn on the camera, Zoom camera. We have a photo session. Untuk semua peserta kuliah Farmasi Bahari, kami mohon untuk mengaktifkan kamera. Kita akan melakukan foto bersama. Baik, kita hitung dari 1, 2, 3. Next slide. 1, 2, 3. Next slide. 1, 2, 3. Last slide. 1, 2, 3. Okay, thank you very much for the time, Prof. Changan Xu and Dr. Eka Prasetya. To all participants, maybe can leave this Zoom meeting we have in the end session. Bagi semua peserta kuliah Farmasi Bahari dapat meninggalkan ruang Zoom karena kuliah sudah berakhir.
Thank you, Prof.
Thank you very much, Prof. Su.
Thank you.
Thank you very much, Prof.
Thank you. See you
Thank
next
very
time.
much for your presentation, Professor Xu.
Yeah, thank you next time. Yeah. See you next time. See you. And thank you. Okay. So I'm leaving. Yeah. And hope to see you again. And also hope to see you in my lab, in my research institute. Okay. See you. Bye bye. Bye bye.